So thanks a lot to the organizers for this invitation, and in particular to Philip to encourage me to talk about phenomics in Genebanks, although we hardly do any phenomics in Genebanks. But I think you're right. There are entry points where phenomics can really contribute to add value to what we are doing and to improve the efficiency with which we are operating. So I'm going to talk a little bit about what we do, and then I'm focusing on where I believe phenomics or high-throughput phenotyping in general can add value and can really change the game we are playing, I believe. So we are part at CIMMYT of this network of CGIAR genebanks, about a dozen of genebanks, mostly in tropical regions, and each one of us conserves usually the largest collection of a particular crop or a few crops. In our case, we conserve the global collections of beans, 38,000, cassava, 6,000, and tropical forages, about 22,000. The cassava collection is conserved in vitro, and the other two collections are conserved as seeds. Over the history of the GeneBank, we have distributed more than 500,000 of samples to more than 140 countries in the world. And actually, when you look at the distribution numbers of the plant treaty, it shows that the CGIAR centers are responsible for the vast majority of germplasm that is being shipped and distributed across borders. Approximately 90% of all samples are coming out of CGIAR genebanks. So here's a brief overview of what I want to talk about. I'm going to first outline, I think, two areas of opportunities for phenomics in genebanks. I think one of them is really to streamline operations, like routine operations, what we have to do anyway. Perhaps we can do it more efficiently. And the second one is really enriching the collection with data, with actionable data for our users that help them to pinpoint the best bet accessions for their particular purposes. And within this value-added data domain, I think there are three different types of data. I'm going to briefly outline those, and then I'm going to enter into just talking about phenotypic data, how I view the division of labor between genebanks and genebank users or collaborators, trade experts. I show you some examples for what we are doing in terms of characterizing the three collections we conserve, and just outline a few practical hurdles in terms of how we could really implement drones, rovers, etc., all these nice, sexy tools in our genebank operations, and end up with one slide about a thought of where perhaps AI is going to change potentially the rules of the game, really, in terms of how phenomics could be deployed in genebanks. So in terms of streamlining genebank operations, I think some of you know Fiona Hay. She's coordinating a community of practice involving all the CGIAR genebanks on seed quality management. And one of the topics there is seed phenotyping. And we have already seen Videometer in a different presentation. We are all the genebanks, or most of the genebanks, are now installing Videometers for seed characterization. Things like length, shape, width, pubescence. We are focusing mostly on color for beans, because this is actually a trait that's important to breeders in terms of targeting particular markets. And we have just started calibrating the machine to tell us which would be the best categories of seed color to describe the different types of beans. There are more opportunities, I think, and the biggest one for us probably is seed sorting. And we don't have an automatic seed sorter. We have about 10 people manually sorting seeds that come in from the field, a lot of manual labor that could be really automated, and we could redeploy that staff to actually characterizing the collections, adding value for users rather than just doing these routine operations. A few years back, we approached a few potential options in terms of providers of seed sorters, and nothing really worked for our purposes, because mostly what's available in the market is for large volume of seeds and a few cultivars, and we have the opposite. We have usually for a bean accession, we have between 1,000 to 2,000 seeds, but we have 38,000 different types of beans, and they look very different. So you have to calibrate each accession separately, which makes it very difficult to implement any sort of automation. At least a few years back, we were told this is very difficult. Also, image analysis of seedlings to quantify germination would be a great tool to have, which we don't have. Again, we're doing everything manually. You can see on the right side all these trays with seedlings. Each of these black trays is one accession, and each of the 38,000 accessions we have to take out every 5 to 10 years to check viability. There's a lot of work that has to be done. If this could be automated, it would be fantastic, but beans are bigger than rice, so there's not so straightforward. And of course, also perhaps multispectral or hyperspectral seed analysis to predict germination, so we don't actually have to germinate. Just run seeds through a machine to predict germination would be great to have. In terms of enriching collection with evaluated data, what we are focusing mostly upon at the moment are genotypic data. Because genotypic data can not only use, we can not only generate genotypic data for our users to sort of help them pinpoint the diversity they're looking for, but also for managing our collections to detect redundancy and so on. So that's very important for us at the moment. We're also focusing quite a lot of efforts to curate the georeferencing data of collection sites so that we can then extract climate, soil, and perhaps disease pressure data from these collection sites into a database that can then be used for a variety of different purposes, for example, environmental GWAS analysis, which I'm going to show you in a moment. But of course, we don't eat genotypes or phenotypes. We don't eat genotypes or environments, we eat phenotypes. Phenotypes is really, at the end, what really matters. And that's, of course, the most complex area. And there are many possible traits. Priorities change over time. Trait expression depends on environment. And of course, complex traits such as, say, drought tolerance are really composed of a variety of different components at different levels, from the metabolome to anatomical features to root system architecture and so on. So it's really the most complex area. So diving briefly into genotypic data, that's the only slide about genotypic data I have here. We basically have 4 use cases where we use genotypic data in the gene bank to characterize the accession diversity, to understand the collection we are holding, to identify gaps, and to identify redundancy in collections. For example, we have found that about half of all cassava accessions are genetically redundant. So there's big opportunity to reduce the size of the collection and again, redeploy funds towards the characterization evaluation of the collection instead. We also use genotypic data to look at how homogeneous or heterogeneous individual accessions are. In the case of beans, most of the 38,000 accessions are land races, and land races often are populations of genetically distinct individuals. So what we are doing there is we are creating pools of DNA samples of about 30 individuals per accession, and then genotype this pooled DNA to identify accessions that are genetically heterogeneous, and at the same time estimate genetic distances among accessions. And we're just starting now to also genotype the cassava collection with a panel of markers that is being widely used by breeders, so that then our colleagues in the cassava breeding program can refer back to the gene bank collection when they are looking for new alleles at certain loci, for example, for the breeding programs. And finally, we are going to use very soon a very low-plex, cheap methodology just for ensuring that during gene bank processes, we maintain the genetic integrity of accessions. We don't mix up accessions, which is always a risk and can never really be avoided. In terms of in terms of environmental data, once we had curated the georeference data of the collection sites, extracted climatic values, we used this data together with genotypic data for an environmental GWAS analysis, both for the cassava and also now the bean collection, to identify environmental footprint of certain, linked to certain environmental parameters such as heat and drought. What we found were several loci where there was a clear relationship between the allelic dose of certain loci, certain genes at certain loci, changing over an environmental gradient, like here temperature and also precipitation. Based on this data, we then went back to the entire collection and pulled out those accessions that had the maximum number of favorable alleles across the loci we identified. And our colleagues in the breeding program are now using those accessions as a starting point for pre-breeding efforts. So now moving on to phenotypic data. I think there is, in general, a sort of division of labor in terms of generating phenotypic data. This is something which seems to be working fairly well in our organization. In the gene bank, we're really interested to characterize as completely as possible entire collections, ideally for inexpensive traits that are generically useful for multiple users. In terms of phenotypes, we really, like all gene banks, focus mostly on morphological and phenological descriptors for identity verification during gene bank conservation processes, not so much as these are usually not traits that breeders are interested in, except, for example, seed color in the case of beans. And if we could add some phenotypic platforms, some phenomics platforms that could collect additional data that could be useful for our typical users, mostly breeding programs, during regeneration of accessions in the field, that would be fantastic. But at the moment, we don't have those tools. And in terms of DNA sequencing, I think there's a clear path towards characterizing entire accessions, eventually at the whole genome level. And as I mentioned, the curation of the environmental data is ongoing as well, and we're starting to also acquire some near-infrared spectra as proxy for, that could hopefully be used as proxy for different traits. In contrast to us, our users are really usually taking very or relatively small subsets to evaluate those subsets for traits of their interest. So what we are usually trying to do is to collaborate with our users so that we work together, and we also eventually receive back the characterization data and can upload them to our database so that the data becomes available to future gene bank users. That doesn't always work, but that's our intention, definitely. We also are starting to try to harvest data from the internet on publications that have used accessions from our collections. So basically what we are ending up with is a sort of a collection of phenotypic data islands for different traits evaluated by different groups of users, underpinned by a sort of sea of genotypic, environmental, and perhaps NIRS data, which eventually can hopefully be used to some extent to predict some of the traits from these genomic and environmental NIRS data. That's our hope. So, moving on to show you some examples of what our gene bank users are looking for in our collections, what they are phenotyping within the organization mostly. Let's have a look here. Yep. So our colleagues in the cassava program are looking in particular, or amongst other traits, but in particular for these traits when they do their initial agronomic evaluation of any materials, early vigor, leaf retention, and early bulking of roots. And they have done a big experiment together with Michael Selvaraj's group, or the Phenomics platform at CIMMYT, to evaluate the core collection, about 500 different accessions in the field that's growing not very far actually from the gene bank. And of course, Michael has all these nice toys, drone-based imaging, LiDAR, and ground-penetrating radar to derive certain parameters that correlate sufficiently well with where you can construct like parameters for early vigor and leaf retention and things like that. Also, we initiated a collaboration with Mineral from Google several years back to test this robotic device, this rover they were developing. We called it Don Roberto, like for rover. And initially we thought, now let's plant gene bank accessions and throw this rover on top of them. But initially we decided to just use like a few contrasting bush bean varieties and evaluate traits that are important for disease resistance and other traits being selected for by breeders. And what we found is that, that the system is actually working quite well in terms of recognizing pods and seeds within pods. The main bottleneck really was that the whole setup had to be very standardized. So it had to be flat land, the soil couldn't be wet, and when it rains, it pours in Cali. So sometimes we couldn't drive the rover at all. And so I think it's a useful system for breeding lines, but for gene bank accessions where you have these tiny bush beans and these huge climbing beans, you can't just use one type of rover. You need something more flexible, or you need different types probably of rovers. And we were going to work on that, but then Google decided to discontinue this mineral effort, and we were given all the images they created, and they are yet to be digested, I understand. Here's another example of what we're doing with the bean collection. We are in the process of, we have to repackage 38,000 seed pouches to essentially move our databases and move from QR codes to RFID tags. And in doing so, we use the opportunity to acquire hyperspectral reflectance spectra, in which we are not trying to calibrate those spectra with different parameters related to the nutritional traits of beans. It's starting to work to a degree, but these are just the very first examples. So I would really like to see whether we can calibrate for protein digestibility, because we know there's a huge variation in the collection between 50 to 90% of the protein being digestible or not. So on top of the protein content, if you can stack on top of that protein digestibility, you can go really very far in terms of increasing the protein content of beans. Another area we are getting involved in, I'm not sure whether you have heard about this initiative, the Periodic Table of Food Initiative, which is a big project funded by the Rockefeller Foundation, essentially a foodomics project. The idea is to go well beyond the typically up to 150 or so compounds that are usually assayed in food. I mean, proteins, carbohydrates, and fats, and within that certain types of fats and so on. But what they are doing is establishing a global network of laboratories to standardize metabolomics assays to essentially identify several thousands of compounds in each of the food samples they are processing. And initially, they're processing about 1,000 types of foods, everything from apple to Indian curry, and beans included. And in the next phase, the idea is then to focus on a few sources of food, and I hope, and I'm pretty sure that beans will be included, to look within these types of foods for the diversity, genetic diversity that could be used for genetic improvement purposes. So this is all in this area of food as a medicine domain that's happening. And finally, here's another example of what we're looking at for the forages collection. This is a new project initiated last year about the problem that the fermentation in the rumen of ruminants of cattle produces a lot of methane, and the Bezos Earth Fund has funded this project to essentially screen the forages collections, not only of Seat, but also Ilri and Icarda, for accessions that, when digested by ruminants, don't generate so much methane because of the presence of certain anti-methanogenic compounds. Tannins, saponins are known, for example, to inhibit methanogenesis in the rumen of cattle. So we are going to screen systematically about 10% of each of these gene bank collections using an in vitro assay to quantify methane production. And what we are screening for here is a low methane production, but high digestibility. So the right lower corner contains accessions of interest to us, which are then subjected to metabolomics analysis to identify eventually, hopefully, some of these anti-methanogenic compounds. Now, some of the hurdles to overcome. I already mentioned that this mineral rover worked to a degree on the main campus where everything was clean and orderly and flat, but we are working in very uneven terrain very often, and particularly for beans, we are working in greenhouses to ensure to control diseases essentially during regeneration. So what would be fantastic to have is a drone that is able to navigate within this greenhouse, knowing the spatial layout of the plots and just flying over each of these plots to identify things like diseases, because as soon as we spot a disease, we need to spray or we need to eliminate the particular accession, but also to monitor growth and to determine phenological parameters such as time to flowering and then time to harvest, pot filling, and so on. It would be fantastic to be able to have a drone that can navigate by itself rather than us manually, because that's just too complex. Now, also a very mundane problem is we're operating in very remote locations. This is one of the sites. We are growing beans in the central Andes, and any high-tech equipment that you leave there overnight is gone the next day, you can be sure. So we need actually to have enclosures to store these high-tech machinery, because it just attracts a lot of attention. And here you can see, again, referring to the rover I showed before, I mean, how do you deal with accessions like that, with bush beans like that, and wild relatives which can grow up to 4 or 5 meters high? Very difficult. And also, I think sometimes, it's funny, but sometimes we have excessive diversity to be able, in terms of mechanizing and standardizing methodologies. Here's an example, here's a photo from one of the forage regeneration plots, and the forages collection has about 700 different species. So very different growth habits, sizes, flowering dates, etc. On the other hand, the advantage of the forages collection is that we're not growing it in greenhouses. So there's room for a drone to fly and, for example, monitor biomass production, which is an important parameter, actually, of course, to our colleagues in the forages program to monitor. So if we could use the regeneration, the seed production for those accessions for the acquisition of like biomass acquisition curves over time, that would be fantastic. And in the case of the bean collection, in the case of the bean collection, I already mentioned our difficulty to find a solution for automatic seed sorting. We were told when we interacted with a group here, not here, in Germany a few years ago, who were developing such a machinery, that the 1,000 to 2,000 seeds that we produce for each of the accessions are just, it's a too small number to calibrate the equipment for that particular type of seeds. But perhaps things have changed. Perhaps there are new algorithms that can do the job. Essentially, what would be great to have is if we could run a particular accession, all the 1,000 to 2,000 seeds once through the machine establishes some criteria, cutoff, etc., and then you run it through again, and you remove seeds that don't fall within the range, the acceptable range, because they are broken, they are infected, etc. So just off-type seeds. That would be really a big help. But so far we haven't found any solution. Now, finally, I think AI, and that's my hope, may change the game. And perhaps I'm dreaming, perhaps that's too far off, and it's definitely not going to happen very soon. But I'm really wondering whether eventually we can get to a point where we can use AI to screen in silico, so to speak, accessions that for a particular trait that the user is interested in, using a multiplicity of data layers, a multiplicity of omics data along the continuum from genotypes to phenotypes, like starting from genotypes to gene expression, metabolomics, perhaps certain anatomical parameters, all the way to growth habits and so on. But for that, we would essentially need for Phenomics to come to the rescue, so to speak, to help, to be able to have methods to screen large parts of the accessions for perhaps some basic sort of component traits that can be stacked on top of each other to create or to contribute to a more complex traits or agronomic performance of an accession under field conditions. I'm wondering whether this is possible. I hope it's not only a dream, but it's going to happen one day. But essentially, really, the idea would be to use these phenotypic data islands to train an algorithm to predict those accessions that cannot be screened for that particular traits from this stack of multi-omic data in the future. Fingers crossed. I don't know. Let's see. And before ending, I would like really to invite people who may be interested to collaborate with us on a range of different topics. And here's my wish list, so to speak. Really sorting out seed sorting would be fantastic. Finding better ways for viability monitoring through NIRS or image analysis to predict or to quantify germination. This could really change the game in terms of how much staff time we have to pay to keep the wheels turning and to continue to conserve the collections, and again, rededicate that staff time to characterizing, evaluating the collections. And also, as I mentioned, like having phenomics platforms to evaluate component traits that may contribute to heat and drought tolerance. The two big traits to screen for climate change. Everyone in our organization and the whole CGR network is targeting climate adaptation. That's the biggest topic, and I think Genebanks can help if we have the proper tools to evaluate and mine the collections. Whenever we mine the collection for a trait, we find a few accessions at least that have the trait we're looking for. So if we had the proper tools available, that would be really fantastic. And again, playing around with AI, perhaps AI can help essentially to narrow down the search space. I'm sure it's not going to be perfect. Perhaps it's just 0.4 correlation, who knows? But anything that can help to narrow down the search space when you deal with a 38,000 accession large bean collection when you search for one trait. All you need is one or two or a few accessions that are really good for the trait. But to get there would be good to be able to extend the scope of what can be evaluated or predicted to a large part of the collection. That would be fantastic. And I'd like to finalize by acknowledging all my colleagues who are contributed, and are going to contribute to what I showed. I mentioned already Fiona, who is managing the seed quality management network. Luis Guillermo, Monica, and Juan Jose are part of the Genebank team, working on bean conservation, forages conservation, and the molecular characterization. Tatiana is working with Darren Drury from Ohio State University on the field spec, the hyperspectral seed reflectance project that I showed you a few graphs of. Jacobo, Alejandra, and Andres are working on the project to identify low methane forages funded by the Bezos Earth Fund. And that's it. Thank you very much for your attention. I'm happy to take any questions.
Peter. Thank you very much, Peter. That was a very enlightening talk to give a little bit of insights into the application of plant phenotyping for Genebanks, besides the traditional applications in conventional plant breeding and in, of course, research. I think that opens our minds to all the further applications that there are and the possibilities that plant phenotyping can open up and enrich. We can have one or two small questions, and the rest then...
The big ones for later?
The big ones for later, exactly. We'll have more time. So are there any questions right now? Immediate questions?
Yeah, thanks. And sure, I'll have a longer question.
Okay. Well, perhaps just one long one instead of two short ones.
All right.
But can you, instead of characterizing the seeds, can you characterize the users of the Genebank?
Can you characterize?
That's not a bit of a joke, but the users of your Genebank.
Mm-hmm.
Can you describe Who are the main
Okay.
users and...
Yeah. The main users are national research organizations in the global south, but we have a lot of users also in the global north, mostly at universities, and also universities in the global south. So these are the the external users. So when we distribute outside of CIMMYT, and internally we distribute a lot to the breeding program. So there's a bean breeding program, a cassava breeding program, and a tropical forages program. And so there's a lot of internal germplasm flow, but the other ones are mostly universities, national research organizations like USDA in the US, for example, as well, and universities.
When looking back at the talks that we heard during this session, were there some aspects mentioned that you would consider of interest now in regards to Genebanks, maybe taking up or putting a focus on coconuts or barley? What was the name again of that little thing on the barley between the ear? Pardon? Peduncle. Maybe peduncle length?
I learned a new word, yeah.
Yeah, me too.
Well, I think the barley example is basically what I would like to see us Genebanks doing as we assemble a new collection. I would love to be able to have it everything like organized like you have organized now in terms of all the data arranged, curated, condensed into Germinate with some satellite databases linked to, and very high passport completeness index, which is for us impossible to achieve because we are sitting on historical data that, you can check to a certain degree, but some things are simply missing and can never be recovered. So that's a huge resource, and it's a huge advantage in terms of targeting traits within the collection, which we don't have in most of the very large collections in the CG centers. And that's the reason why it's so important to characterize and evaluate those collections much more deeply. Because when I look at our bean collection, there are thousands of accessions which have a number and that's it, for which we sometimes even are not completely sure about what kind of adaptation, what kind of growth habit they may show. So we need to take them to different sites. We have multiple sites of multiple elevations in the Andes to regenerate differently adapted bean accessions. And sometimes it's just trial and error because we just don't know almost anything about those accessions. So yeah, this is such a great resource. I was very impressed.
Peter, with the genotyping becoming more affordable and with a lot of the Genebanks now routinely, running SNP data across entire collections, do you think it'll be easier to sort of put together those panels that represent a large proportion of the diversity that could, in a sense, then become a resource for the community so that, you could potentially, or I guess the community could search for funding to then grow those lines and invite people to come and phenotype for their trait of interest?
Yeah, your model of community-based phenotyping is fantastic. And we had a brief conversation, I think yesterday or the day before. It's a great model. We would love to do something similar in Colombia, but we don't have so many research groups in Colombia focused on beans, for example. So it would require international travel and everything, so it becomes more complex. Yes, essentially, yes. Our strategy is to first genotype, ideally the whole collection. We have finished cassava. We are lacking still resources to do the other two. I think we probably will have soon 10 to 12,000 accessions, bean accessions genotyped, but there are 38,000. There's more required. So then initially, putting a few tens of thousands of markers, ideally with a methodology that minimizes ascertainment bias, to describe diversity and then condense into a subset, a core collection, and which probably should be whole genome sequenced then and become a resource for the community. Erie for rice did something similar. They didn't genotype the whole 120,000 accessions. They selected a subset of 3,000 and whole genome sequenced them straight away. And as soon as those sequences became available, suddenly everyone wanted to phenotype those 3,000, which is, so it creates a community dynamics that leads to stacking of traits being characterized for those subsets. So I think that's definitely a way to go. And as you stack traits on the subset, if that subset is cleverly chosen, I think you can then use anything that can be learned from machine learning to extend and predict similar traits in the non-characterized but genotyped accessions as well. So to sort of spread the knowledge around as much as possible.
Here I am.
More questions?
Yeah, of course. I've been visiting several gene banks worldwide. Japan, Bosnia, of course, and Belgium. Is there some trends that you mentioned this community interaction between gene banks to become more automated, become more data-driven? Did you see some developments past years?
Yeah, I mean, this is something I'm hoping for, waiting for, and trying to contribute to. Amongst the CGR gene banks over the last 1.5 decades, we were mostly occupied with getting our house in order, to be frank. Because we, like 1.5 decades ago, we came, or the various CGR gene banks came out of a period of chronic underfunding. And there was a huge backlog of accessions that required urgent regeneration. So really, during the last 15 years or so, we were just maximizing the numbers of accessions to be generated just to keep the collections alive. And we're sort of getting to the end of that tunnel, so to speak. And now I think there is clearly a movement towards well, now that we have secured the conservation, let's try to make it easier to use those collections by creating value-added data, right? Automation, to a degree, I think, I think automation, the global leaders in automation, gene banks are probably in Japan and China, Korea, not CG, not other national gene banks so much.
No, that's right.
So we have a lot to learn from them, I think, from those. I remember I haven't been there, but in Japan, the gene bank is everything. You don't go into the cold room and retrieve an accession. You push a few buttons on a screen, and the robot arms go in and take out what you need.
I was there. I made a video.
Are you over there? Okay.
I can show you later. It is fascinating, actually.
Yeah.
Another question, actually, we have a cool project that's about exploiting crop wild relatives in breeding programs. We've been running some trials with multiple crops. But the crop wild relatives are nasty plants to grow. They are wild. I would like to hear is that a common issue that you are struggling with, that they are difficult to phenotype, basically?
They are bloody difficult all the way around. Yes. And actually, I mean, in terms of conservation costs, it's probably each accession of a crop wild relative is at least 5 to 10 times as expensive to conserve as a cultivated counterpart usually. I mean, crop wild relatives are of great value if breeders run out of options, particularly disease resistance, for example, right? But they are very expensive and difficult to conserve, and even more difficult to characterize at a phenotypic level. Genotypically, it doesn't really matter. As soon as you can get out clean DNA, it doesn't matter. But phenotypically, yeah, it is big headache.
Yeah.
And I mean, I've shown one of the photos of a guy walking in the screen house, surrounded by this huge canopy of climbing beans. These were wild. I think these were probably wild relatives because of the station where they were grown. And yeah, I mean, in the CG, we have to fulfill certain quality or KPI standards to qualify for ongoing funding from the Global Crop Diversity Trust. And one of these KPIs is that 90% of all of the accessions we are conserving are available for immediate distribution globally, meaning that have been regenerated in sufficient numbers and have been tested, in our case, for about 40 different diseases each seed lot to ensure that they're clean and can be distributed. To get to those 90%, most of us were focusing on the lead material, or the primary gene pool, but not the secondary and tertiary gene pool, because to produce seeds of wild bean species, it takes 2 to 3 years for a good part of the species, whilst for Phaseolus vulgaris, in landrace takes you 3 months. So there's a huge difference.
Yeah. Okay. Yeah. Thanks.
You worry.
Any further questions? John?
Hi. I had a question about how you. I'm curious to know how you think about rapidly changing sequencing technology, and so the balance between storing sequence data now and how easy it's becoming, and like the advent of long read sequencing. I'd be curious to hear you just talking a bit more about that. Like, we know structural variants are important for traits, and up until recently, it was hard to get long reads for as cheaply as it is now. So,
no, we're not into long reads yet, because we're not really going for resequencing whole genomes, etc. But actually, most of the CGI gene banks have done is to settle on one methodology, so we can also compare collections. For example, we are conserving cassava. IIT is also conserving cassava. We use the single methodology to genotype and compare both collections. And in terms of structural variants, what we found was that we're using the DivSeek technology, which produces, essentially it's a reduced representation sequencing approach that produces both SNP data and also presence-absent variation data, essentially. And we always find, as soon as we step out of a single species and compare multiple species, these structural variants, of course, become very important for distinguishing, for estimating genetic distances among accessions. But again, few of us have reached the point where we're starting to think about long reads and whole genome resequencing of large proportions of our collections, except IRI, who have whole genome sequenced several years ago already, 3,000 rice genomes. But they have the advantage of dealing with a relatively small genome. I mean, our colleagues in CIMMYT have a much bigger headache with their wheat collection of decent size. You have a follow-up question?
No, but that was really interesting. But I guess I've heard someone say to me once that the best way to store DNA is in a cell that you keep intact. And so in that sense, like if you can keep, like, you've got the seeds, is it worth? The seeds are being stored, and that's pretty permanent. It seems like they could be really good resources just for, as questions come up, you've got the seeds and you can use the best sequencing technology of the time.
Yeah. I mean, so storing seeds versus I think that they essentially, several years ago, we were dealing with RFLPs and RAPIDs and AFLPs. Those platforms are not really future-proof or forward-compatible. As soon as you have at least sequence tags, you will be able eventually to integrate that into whole genome sequences as they become available. So in that sense, I think as long as you're sticking to sequence-based genotyping technologies, which everyone is doing today, essentially, I think you have a degree of future-proofness built into the system, fortunately. I was going to mention something else, but just escaped.
That's okay, Peter. The time is up, and I would like...
The beer is ready?
Yeah, the buses are getting ready, and I think you should do too in order to attend the conference dinner. I would like to thank Peter one more time. Thank you for being here.
Thanks for the invitation.